Hello people. Haematopoiesis is the formation of blood cellular components. And haematopoiesis are actually coming off from one cell. And this cell is called the pluripotential hematopoietic stem cell. So all blood components, blood cellular components that are circulating in our blood come from this one hematopoietic stem cell. And before we see how these lines of differentiation are going, we will analyze what actually is, where is this hematopoiesis happening, in which organ. So there are two parts of our life. One part is the part in the uterus, our intrauterine life, and this hematopoiesis is called prenatal. So prenatal hematopoiesis, and the other part of hematopoiesis is happening after we are born, and this hematopoiesis is called postnatal. And we can see that prenatally there are a few organs that produce hematopoietic cells and that do this job of hematopoiesis, and after birth, so in our adult years of life, hematopoiesis is happening only dominantly in one organ physiologically. It can pathophysiologically happen in other organs. So after our intrauterine life begins, the first organ that produces cells resembling erythrocytes or hematopoietic stem cells, erythroblast cells, is the yolk sac, and that is happening somewhere 3 weeks into intrauterine development. So the yolk sac is the first that produces cells, and that actually functions as an organ that produces of hematopoiesis. So this phase where the yolk sac is producing cells of the blood is called mesoblastic phase. So it is called mesoblastic phase. The second organ that begins with production of blood cellular components at 6 weeks of life is actually the liver. And the liver, when it starts producing, this phase is called the hepatic phase. The second organ that also produces with the liver is the spleen. This phase is called the lienal phase. So we can see that these two phases overlap, but the liver and the hepatic phase is the dominant organ in these months of intrauterine life that produces blood cellular components. Somewhere at 6 months of intrauterine life, the bone marrow is starting to become the main player in producing blood cellular components. And this phase is called myeloid or medullary phase. So the medullary phase. When we are born, we can appreciate that the bone marrow is the main organ producing our blood cellular components. But the bone marrow has two parts. We have in our body two types of bone marrow. One is the red bone marrow, and the other is yellow bone marrow. So basically yellow bone marrow helps store fat, but the red bone marrow is where the hematopoiesis is happening, and it produces the blood cellular components. The red bone marrow in an adult human is not present in every bone, but it is present in some special types of bones, like in the skull, in the vertebrae, in the sternum, in the ribs, then also in the pelvic bones, and it is present in the end or in the epiphysis of the humerus of our long bones, the humerus, the femur, and the tibia. So these bones have this red bone marrow that produces our blood cellular components. Now, we will go back and see how these cells are actually produced in our bone marrow. And the process is relatively complicated. The first cell that we can appreciate is the pluripotential hematopoietic stem cell. So the pluripotential hematopoietic stem cell, it is a mouthful. This cell has the capacity of asymmetric division. It means it will produce another cell with different characteristics, but it will produce a cell that is the exact copy of itself. So it is another stem cell of its type, and thus replenish its cell population. But it will also give rise to cells with different characteristics. So this cell in this hematopoietic process gives rise to two multipotential stem cells. One is called CFU-GEMM. So this actually means colony forming unit, means it can form these colonies of cells. Granulocyte, erythrocyte, megakaryocyte, and monocyte. Actually, this stem cell, can produce all of the granulocytes, erythrocytes, and platelets in the end. So this is the first cell coming from the pluripotential stem cell. The other cell is called colony forming unit cell lymphocyte. So from this cell we have the T and B lymphocytes arising. These cells in this upper quadrant are called stem cells. And obviously they have a high potential of differentiating in all of these other cells. Now we will forget this lymphocyte line and we will concentrate on this part of the hematopoiesis. So these cells in this second quadrant are actually called progenitor cells. The colony forming unit cell, granulocyte, erythrocyte, megakaryocyte, and monocyte gives rise to other progenitor colony forming unit cells. So this first is the colony forming unit for erythrocytes, thus E. Then the second one is the colony forming unit eosinophil, and this is the colony forming unit basophil, and this is called the colony forming unit megakaryocyte. Then we see that we have an intermediate cell, but it is also a progenitor cell. It is called colony forming unit GM. It produces this colony forming unit cell G, means granulocyte, but it produces neutrophils. And this is colony forming unit M for monocytes, because we have this line that produces monocytes. So these progenitor cells, we see that in the end, this makes it a little bit complicated, this intermediate cell, but in the end we got the erythrocyte cell that is forming the units of the erythrocyte, the granulocyte line with the eosinophils, basophils, and neutrophils, then the platelet line with the megakaryocytes, and the monocyte line. So everything here is producing everything except the lymphocytes. So we will see how further these cells are differentiating. This third quadrant, where we have these cells are called precursor cells. Precursor cells. So if we look at the erythrocyte line of cells, the first cell always has the blast in it. So the first cell here in the erythrocyte line is called pro-erythroblast. So pro-erythroblast. Then this cell appears a little bluish because it has ribosomes in it. Then with more ribosomes, it has this more bluish color. Then it is called basophilic erythroblast. The second cell has a little bit of ribosomes and is producing also a little bit of heme, thus having both a little bluish and a little bit of red color. Appearing with more colors, it has the name polychromatophilic. So polychromatophilic erythroblast. The last cell has a lot of hemoglobin, thus it becomes reddish, and it is called acidophilic erythroblast. This cell it loses its nucleus and becomes a reticulocyte. The reticulocyte then goes into the bloodstream, so all these cells that are in the lower quadrant are mature cells. These cells are present in the blood, but the reticulocyte also, it is in the bone marrow, but it enters into the blood. And in the blood, it becomes a mature erythrocyte or a red blood cell. So we see how these cells are progressing in this line to the erythrocyte. Then the process is similar in all of these three lines. So we have here, this line produces eosinophils. This line produces basophils, and this line produces neutrophils. They have the same names of their precursor cells. You only put the prefix of the end cell. So the first cell is called the myeloblast. So this is the first cell, and it is called the similar in all of these three lines of cells. Then the second cell is called promyelocyte. The promyelocyte, and this is the same here and the same here. But only this would be the eosinophilic promyelocyte, this would be the basophilic promyelocyte, and this the neutrophilic. Then this cell is called myelocyte. And the last cell is called metamyelocyte. And it is the same in all of these groups of cells, only the prefix is actually different. Before these cells go into the blood, there are also some band forms. So the nucleus isn't really segmented, and in the blood it becomes actually the mature cell that we know. Then we have this line producing platelets. The first cell is, of course, the blast. It is called the megakaryoblast. So all these first precursor cells have this blast in them. The second cell is called promegakaryocyte. Promegakaryocyte. From this promegakaryocyte, we have this cell called the megakaryocyte. This megakaryocyte has these platelet ribbons that are going into our blood vessels. And from these platelets ribbons, one by one, it releases our platelets. So this is how platelets are being released into the blood. The last of this part of the haematopoiesis are the monocytes. And the first cell is, of course, a blast, the monoblast. The second cell is called promonocyte. site. And then the last cell is the mature cell, the monocyte. So this is actually how all of these cells are being produced. We have the lymphocyte part left. So the lymphocytes are being produced from their stem cell, the colony-forming unit lymphocyte. It gives rise to these two cells, the colony-forming unit cell for T lymphocytes and the colony-forming unit cell for B lymphocytes. Of course, the first precursor cell has a blast in its name, so this is the T lymphoblast, and this is the B lymphoblast. The last cells are, of course, T lymphocytes, naive T lymphocytes, and B lymphocytes. These cells are special because they need further education. For example, the T lymphocytes are being further matured in the organ called thymus, that is not in the bone marrow, but somewhere else in our body. So basically, this is how these cells are maturing. And this is a simple representation. Between all these steps, there are specific cytokines and a specific microenvironment that is helping these cells to mature this way and help them that there are no problems that are arising in their production. So we will now see how the maturement of erythrocyte happens, but very clearly and detailed. So in the bone marrow, there are macrophages present. These macrophages help these cells to mature. So these cells are the erythrocyte precursor cells. And this is the reticulocyte. This big cell is a macrophage. So this is a macrophage. And this macrophage helps these cells to mature. It gives them iron and it produces cytokines to help them to know in which direction they should mature. So this special construct that is present in our bone marrow is called the Erythroblastic Island. And from this cell, the pro-erythroblast, to a reticulocyte, it takes about 7 days to produce something like this. And the process is going, so we have the first cell. The first cell is the pro-erythroblast. So the pro-erythroblast is a big cell with a big nucleus and with some organelles. And then we have, it matures further into the basophilic erythroblast. The basophilic erythroblast has some ribosomes that are starting to produce proteins. Then it matures further into this polychromatophilic erythroblast. The polychromatophilic erythroblast has these granules. These granules are called siderosomes. The siderosomes contain aggregates of hemosiderin, and hemosiderin is nothing else than an iron storage complex. So it stores iron. Iron is an important component of hemoglobin. In this cell, ribosomes are starting to produce globin, and mitochondria are starting to produce heme in order to make hemoglobin. These cells further mature into this cell, and this cell is called the acidophilic erythroblast. The acidophilic erythroblast is coloring itself red because it has a lot of hemoglobin in its cytoplasm. In the end, the macrophage actually devours the nucleus of this acidophilic erythroblast, and it becomes the reticulocyte we see here. And the macrophage is guiding the reticulocyte to the blood vessel that is here, the blood vessel, and the reticulocyte is going into the blood, further maturing into the erythrocyte. So this is actually the complex microenvironment where a lot of cytokines are being secreted and produced in order to help the erythrocyte to mature. I will only mention one important cytokine, and this cytokine is actually being produced in the kidney. And if we have hypoxia, cellular hypoxia in the kidney responds in production of erythropoietin. Erythropoietin is a cytokine that acts primarily on this cell. So it acts on this cell, erythropoietin, and it helps this cell to proliferate. And if this cell proliferates, of course, we will have many erythrocytes. So we see that this hematopoiesis is a complex lecture, and it is a complex theme. So this is a simple and easy representation of a physiological process called hematopoiesis. Thank you.